we are getting forward with our talks on hyperscanning. So it's my great pleasure to be here with Guillaume Dumas.
Hi, everybody.
Unfortunately, we are still pretty much locked up, as Guillaume and I just discussed before, but we're going to focus on some brighter topics today. But let's go orderly with everything. I'm just going to start sharing my screen. So the topic of our today's talk is hyperscanning, history, hypes, and hopes. And I think that the title itself is very interesting, at least for me, and I'm very much looking forward to what I'm going to hear. My name is Ivan Gligorevich. I'm from MBT. I'm going to be your host without further words on myself. Just focusing on a few things before we start. The goal of this talk, for those of you that maybe listened to the previous talks on hyperscanning, we had a chance to dive into some studies that were very interesting, done, for instance, by Suzanne Dicker and Martin Imhoff and Karl Philipp Flosch. And we also had the chance to talk to Francisco Parada and Martin Bleichner. So we had the chance to see parts, bits and pieces of hyperscanning effort. But now we're going to try to zoom out and try to observe the big picture and see where did it start, what motivated this whole field, what are the current promises, and what are the hypes, and where it is all going. We are going to keep to 90 minutes, so 60 plus optional 30. And I encourage you all to ask questions in the question tab anytime. And I apologize in advance if the answers are not timely or we don't have the time to cover everything up. So, but we'll definitely try to answer most of them in discussion part of this talk. We will record this video, and highlights will be posted on YouTube. Maybe we'll cut some interesting answers and to make it easier to find. So we will see about that. As a sort of introduction, I don't know how to put in a few words what Guillaume has done and is currently doing, but I certainly enjoyed reading some of his papers, at least in the EEG domain that I understand much more than the other part that he's dealing with, because he's also in the biology and is a proponent of these closed loops that actually affect everything. And I'm sure that he's going to cover some of the talk. He's working currently in Institut Pasteur, but as I understand, he's soon to be expected in Montreal as well. So he's working on the embodied and reciprocal nature of human cognition with behavioral and biological and social levels of interaction. And for those of you who haven't read his famous papers from 2010, 12, 14, 18, I would strongly encourage you to do so. And also visit his website. Guillaume, before I give the floor to you, can I ask you to sort of in a nutshell tell us why should we do hyperscanning, and what is it that you try to achieve with the super complex hyperscanning setup from 2010 that would justify this entire effort that you have done?
Oops, sorry, I was muted. Yeah, well, that's part of the things I want to cover, because I think the big question and the hype part of the talk is, do you really need hyperscanning, and what kind of question need hyperscanning, and what kind of question doesn't necessarily need it? And in the case of 2010, I would use this example to explain what was the core question we wanted to answer from a scientific point of view, which was like, to what extent our perception action loop between each other allow our physiological system to get entrained at the very fine-grained temporal dynamics. And why? Because there are like physiological phenomena such as plasticity that occur at those millisecond time scale for learning, and our neural networks learn to match representation for self and others. And so having a learning of those self and other patterns required to get this fine-grained timing. And so like that was one of the motivation, and the other motivation was more like deeply rooted in history of science and embodied cognition, because I had like the chance to do my PhD in the former lab of Francisco Varela, who developed all these embodied cognition and active account of human mind. And among the last contribution, the embodiment aspect was not only about the body of the person, but also the radical embodiment with the social realm, and how we should adopt cognition not only from an individual point of view, but as Varela was putting it in his last works, consciousness and cognition is a public affair, and how we have to bring back the social dimension in the study of human cognition. So like how social dynamics and social interaction are actually constitutive of our individual cognition. And that's kind of problematic, and there are a lot of people who would not necessarily agree with that, that you need to account social interaction as constitutive of individual cognition. But in order to probe that, you need to have dyadic recording and to have this kind of hyperscanning systems. So that's the two main motivation: physiology and cognitive science and embodied cognition.
Okay, I'm very much looking forward to what you're going to say. And I'm going to stop my screen sharing now and give the floor to you.
Okay, so let me share screen. Oh. Okay. Can you see the slide?
Not the slide yet, but I see the black screen. So
is it good now?
Oh yeah, now I see a slide. I'm going to.
Okay.
Yeah.
So so as the title put it, I'm gonna try in the last in a short amount of time to cover a brief history of hyperscanning, the hypes and the hopes also, because I don't want to finish on the negative aspect about that. So as I say, like one of the key motivation was clearly the place of social interaction in cognitive science, and how over the last decade, social neuroscience realized that we have missed paradoxically a big aspect of social cognition in human, which is the interactive context. And so that's very well covered by, for instance, in the case of Pfeiffer and colleagues, as metaphorically the dark matter of neuroscience is this social interaction. And at that time, when you were looking at the publications of two individuals engaged in dynamic interaction, there were not much, and even less regarding multiple individuals beyond the dyad. And over the last 10 years, I tried to use hyperscanning, but other approach to naturalize social interaction and better understand the neural underpinning of cognition in the social context. And so like this, like the simplified research program with either brain with brains, models with models, or brain with models. And so today I'm going to focus, of course, with brain with brains in hyperscanning EEG. But interestingly, the models with models also can give us clue about the underlying mechanisms of the strange phenomenon of interbrain synchronization. And as you say, like I'm going to move soon to Montreal to create my own lab, the Precision Psychiatry and Social Physiology Lab, where the goal is going to be to combine this interactive, social interactive task from integrative neuroscience with computational biology to better understand neurodevelopmental disorders, especially autism, through this kind of systemic physiological approach of social interaction and towards precision psychiatry. And for that, I'm putting social interaction within the different dimensions that are to take into account in physiopathology. So from genetics to social interaction. But so that's more like a context. So now let's start for history of hyperscanning. So if you look at hyperscanning on the internet, and if you have like Google alarms to tell you what are the news in hyperscanning, you may know that hyperscanning is in the first place the name of a Mattel video game console. So I won't speak about that, but it's funny to know that. Hyperscanning is a term coined by Reid Montague, one of the pioneer of computational psychiatry as well, actually, where in 2002, he clearly stated in a neuroimage paper that if you have sufficient funding to get multiple MRI and you are able to synchronize those different MRI together, then you can do what he called hyperscanning, which is recording of different brain activities simultaneously during social exchange. And actually, it's funny because in those videos, it goes even beyond the diet. So if you have really a lot of money, you can have many MRI machine and do a group hyperscanning. I will go back to that at the end for the hopes and for instance, the work of Susan Dicker on group hyperscanning in EEG. But why not? You can do it also with MRI. But so also in the early paper of Reid Montague, an interesting aspect that was pretty visionary is that beyond just synchronizing recording of biological and physiological activity, we can also couple and synchronize those channels. So having the physiological recording affecting the behavior and vice versa. So even kind of foreseeing the use of hyperscanning in a neurofeedback type of context, which is not something yet achieved. And if you look at the publications that used hyperscanning or multi-subject recording in brain activity, well, actually it doesn't start in 2002, but in 1965. If you do your homework and dig into the literature, in 1965, there were like a science paper by two ophthalmologists, Dumas and Berend, about hyperscanning in a very strange context. So it's funny to know that Science was publishing this kind of paper in '65 because these two scientists pretend to have found at that time a telepathic communication between identical twins with just one figure. So here you see the trace. That was the only figure of the paper. Of course, that's never been replicated. And I suppose that even this kind of parapsychology focus initially prevent people to do hyperscanning afterwards, and this kind of multi-subject recording, because it was kind of associated with these weird parapsychological topics. But so in 2002, when Reid Montague come back with hyperscanning, It's with MRI and with the idea of computational psychiatry and the study of decision making in neuroeconomy. And quickly, there have been nice papers in big journals about how hyperscanning recording can decipher decision making in this social context. So, for instance, the work of Kim Casas has been one of the first one. And simultaneously, other type of study popped up, not yet in EEG, but in fMRI. For instance, here in the case of Christian Kaiser with Marlene Schiepers, there have been like study of charade and how to transmit information from one brain to another, or with this work from Uriason in Princeton about neurocinematics and people watching the same movie. In those two cases, I won't call it, strictly speaking, hyperscanning because the people are not simultaneously recorded. But it's interesting to note those studies because they are kind of also in air du temps, in the contemporary topics, about recording multiple people and looking at more natural stimuli instead of just Gabor patches. And in the case of the work of Uriason, also an interesting aspect for social neuroscientists is that the peaks of correlation between brain activity when they were watching the good, the bad, and the ugly, for instance, were during the social scenes. So that's the brain intrinsic neural activity of those different people tend to be more similar during context of social interaction. That's for MRI and fMRI because hyperscanning, the scanning aspects come from that. There has been other techniques, of course, that use this idea of recording multiple brains simultaneously. In fNIRS, there are many, and also focus a lot on neuroeconomy initially and collective decision making. But if we go to EEG, it's the team of Fabio Babiloni in Roma who really pioneered the field of hyperscanning EEG with the most hyper paper in history with Hypermethods for EEG hyperscanning, where they recorded people playing Trete, a card game, and as a proof of concept show that coherence between brain's activity of people playing in the same team were more similar than between brains of the two people in different teams. On the other side of the Atlantic, where actually I did my postdoc later on, the team of Scott Kelso with the pioneering work of Emmanuel Tognoli and Julien Lagarde study hyperscanning EEG to have more. So like here we go back to the question, why are you using hyperscanning in the first place? So here it was for investigating this perception action loop in dyads. They wanted to go to the interbrain level, and actually I would show that there were like preliminary results on that. But what they uncovered is especially intrabrain neuromarker they called Phi over the right parietal, which was facilitated by this active social interaction compared to more like passive spectatorial social perception. So this is more like at intrabrain level, but they also digged into the interbrain level, and that's why it's always good to go to poster session, because in one poster in SFN 2005, they talk about this idiosyncratic pattern of interbrain synchronization that they observe in their task, especially during this perception action loop between frontal area of the leader, the person who do the movement, and the more parietal and occipital region of the follower that imitate the leader. And that was not yet full bulletproof statistics, but already Isaac Asimov say like the most important things to hear in science is not necessarily eureka, but that's funny. And these idiosyncratic patterns were like, they're like, huh, there is something going on here. For the statistics, we have to wait 2009 with Andres Linderberger group in Berlin, who using a very constrained but very controlled setting, managed to prove that statistically speaking, there were like these bursts of interbrain synchronization in theta band between these guitarists playing together with the same metronome. And so that was interesting because it was the first real statistical demonstration in EEG of ongoing interbrain synchronization. In the case of the work of Fabio Babiglioni that I showed before, it was average across the whole trial. Here we go back to the more like ongoing interaction, but still they were like this metronome. And so to summarize, we've seen that hyperscanning initially showed that we should move from social perception to social interaction, and that actually there is anatomical functional correlation across several subjects when they are immersed in this naturalistic stimuli. So that's more like the work of Uriason. That there is dependence of information contained in the EEG signal, and that can be measured and tracked to activity up to the gamma band. So like this is more like what Fabio Babiglioni showed with the card game recordings. It was like sequential recording and not the real-time track like in the Lindenberger. For the dynamics, the work of Emmanuel Tognoli and colleagues showed that there were dynamical neuromarker of motor coordination, but it was intra-brain, and the work of Lindenberger in Berlin showed that you can have synchronization and they can be observed and quantified statistically, but still in that case, it was with an external metronome. And so that was this combination of aspects that motivate the study in 2010, plus the inspiration from developmental psychology, who was doing behavioral hyperscanning, so to say, because developmental psychologists for decades have recorded interaction between babies and their parents. For instance, my physical supervisor, Jacqueline Nadel, at that time, she had like studied a lot how a 3-month-old infant can decipher the difference between the ongoing video of its mom or a pre-recorded video of its mom. So we have already at early age this ability to detect fine-grained dynamical contingency in social interaction. And inspired by that, we did like this system, fairly complex indeed technically at that time, with 2 Faraday cages, two video in analog recording to have no delay between the two, the camera and the video. So that's also something we discussed in earlier webinar, but the timing is very important in hyperscanning. And so the subject were seeing each other through this video system. And here is a representation of what they were like doing. So initially a baseline with no view, no motion, so NV and M. That's kind of the ultimate baseline where brains are at rest and not doing the same task. Then another condition arrive. Oh, my computer is lagging a bit. I hope you see the video properly. So the second baseline was no view motion. So it means like participants are moving their hands, but they are not seeing each other. And here you see now that there is an indexation at the bottom of the screen, which indicates synchrony, imitation, and drive, and that's actually the interactive part. So I think my computer is crashing right now. No, I hope. So,
yeah, you're just breaking up a bit, and the video is not super smooth. But I think the point is there. I hope the computer doesn't crash completely.
yeah, I'm trying to. Maybe that's the video that's really put like a zoom on top. Let me see if I stop zoom from now. Oops, sorry. So my presentation crashed. Sorry about that. If there are like maybe intermediate question to while I'm restarting the thing, but so as you saw, the motivation for instance, this 2010 study was really to connect those existing pieces of information. We know that there are like anatomofunctional similarity. We know that there are fine-grained dynamics that can be shared. But to what extent we can achieve that in a pure reciprocal setting without external metronome and without having to average across the whole interaction. So like during the ongoing interaction. So I'm restarting the thing. Up. Okay. Back on track. I think like the Zoom plus the video was too much video at once.
Yeah, that happens.
So can you see the slide again? Yes?
Yeah, yes, we can.
So even before going to the interbrain level, at the intrabrain, so like in the work of Emmanuel Tognoli, we also took the advantage of having this complex system with 2 Faraday cages to have a gradient of tasks ranging from doing nothing, doing the same thing, like moving the hands without seeing each other, then imitating in one direction, so subject 1 imitates subject 2, and then having the real spontaneous one, so like subject 1 and subject 2 move their hands and they imitate each other whenever they want. And interestingly, there is this turn-taking that emerged that was in the video. I hope you catch a bit this phenomenon of turn-taking that appear, because it's kind of nice to see that even at the nonverbal level, you have this regulation of dynamics that occur. And so first thing first, how the brain react in those different contexts. And interestingly, there are different neural correlates depending on the context. So if you are forced to imitate someone else, or if you are imitating someone else in a spontaneous context, the neural correlates of those imitations are different. So that's one first difference. And the other one is, of course, the role that you are taking. So even if you do exactly the same movement at the same time, if you're doing it while being the one that is leading or the one that is imitating, even from the external point of view, you seem to see the exact same behavior. Well, the brain propensity is not the same. So like the stance you have is not the same. And so we show that there are different neural correlates of those different context and role. And interestingly, thanks to the team of Philippe Fossati at la Salpêtrière, we even show that in fMRI, we can retrieve those dissociation. And something that we cannot see in EEG that can be seen in fMRI is that there is, for instance, an intrinsic reward of being in the spontaneous interaction that is not there during the forced imitation. So that, that's one of the differences that you can have between contexts. But so since we are more like interested in interbrain and hyperscanning, so here the idea was to have this fine-grained behavioral indexing of the ongoing interaction and combine those behavioral analysis with EEG analysis. So taking activity of the sensor of the two brains, filtering it, extracting the phase, and trying to quantify objectively the synchronization between those signals. We were able to recreate this synchronization within brain and between brain and look at how they unfold during the interaction. So here on the top left, you can see the matrix of connectivity with subject 1 and subject 2. And it's like a battle chip. So you have one row is one sensor and one column is one sensor. And you have like subject 1 with subject 1 on the upper left, subject 2 with subject 2 on the bottom right. And the interbrain part is on the top right. And you can see on the histogram on the right that are moving. In green, it's the phase locking value, so the synchronization within brain. And in blue, those are like the hyper phase locking values, so the between brains. And the first observation is that it's non-Gaussian and
Guillaume,
that the between brains are not strong.
Sorry for interrupting you.
Sorry.
You were cutting while explaining the histogram. Can you maybe repeat that part? The green and the blue line.
Yeah. So the histograms are showing intrabrain and interbrain synchronization value, and the two first message we can get is that first intrabrain are stronger than interbrain. Well, that you are more synchronized with yourself than with others. That's reassuring in a way. And the second thing is that it's non-Gaussian. So it needs to have proper statistics that handle that. So in the case of phase locking value, you need to use non-parametric statistics. And so for that, at that time, I took inspiration from the pioneering work of Eric Maris and Robert Ossenfeld with clustering statistics to try to have the most robust and still not too much conservative thresholding to see what are the synchronization at the interbrain level that are statistically significant. And that ended up with these results where we found that indeed in different frequency band, alpha, beta, and gamma, there is this synchronization between the model and the imitator. And something that people not necessarily catch in these results is that it's symmetrical in the low frequency, but not symmetrical in the high frequency. So in the more you go in beta and gamma, the more it go frontal in the model and occipital, parietal in the imitator. So kind of align with the idiosyncratic patterns that Tonioli and colleagues were speaking in their SFN poster. But still, in 2010, it was not really established to have those kind of analysis, and it was a real pain to get published. And I had to demonstrate it in a second different way. So that's also using a permutation test, so with bootstrapping. And the idea here was to bootstrap the behavioral analysis and to look at to what extent what we observe as synchrony between brains is collapsing when you scramble the behavior. So that's actually one interesting approach in hyperscanning to know if what you're observing is robust, is to just take exactly the same analysis pipeline and scramble the behavioral analysis. And if it survives, then it means that there is something fishy here. And fortunately, in our case, we managed to replicate with this surrogate approach. So indeed, it was dropping back to 0 when we were cutting the behavior. So that's more like historical aspect. Actually, let me see. I wanted to mention, I don't know if the person is in the thing, but there is a recent review in frontiers about hyperscanning that I recommend to have like a up-to-date review. And now I'm gonna switch to hypes and hopes. So for the hypes, I would say that from what I told about the pain, it was to publish the 2010 paper, and it ended up in +1 at that time. It's funny now to get published in bigger outlets, because now it's getting hyped, not necessarily in a bad way. So we had like this wonderful paper with Shametsori team with Pavel Goldstein in PNAS, and actually in 2010 PNAS refused my interbrain paper. So it's funny 8 years later to go back to them. That show how the context has changed. And so now, instead of having reviewer telling you are a parapsychologist, you can go to big journals and make covers with the results. So it showed that it's not necessarily a negative comment, but it showed that things are moving and changing in the apprehension of what those results are. And maybe it can go further than what a scientific conclusion can be done. So for instance, there's been this huge hype about the tech-lepathy, so telepathy based on interbrain recordings. So this is interesting from a technological point of view and as a proof of concept, but that's where the hype start to be concerning to me, because people overinterpret those proof of concept at the technological level and go beyond the scientific conclusions. And I would say that for a good reviewer to comment that helps to get a sense of if you need hyperscanning or not, is that can you do it without hyperscanning? So if your scientific question can be answered without hyperscanning, then it means you are doing hyperscanning just for hype and not for a scientific question. So I think that's a very simple way to probe yourself. Can you try to decipher and answer your scientific question without hyperscanning? If yes, actually it's even not worth the pain, because it's a pain to do hyperscanning. So yeah. For the hopes, well, I briefly mentioned that actually Reid Montague was already mentioning the coupling between behavioral channel and biometric channel. And I think the hopes in the futures of hyperscanning is to integrate neurofeedback and to have like hyperscanning combined with feedback to the subjects. Especially now I'm interested with psychopathology and conditions like autism, where social interaction can be challenging. So to have such feedback can be useful. Another hope is to not have, again, to do a frame-by-frame analysis of behavior, because what I show you for the 2010 paper took 11 months. I was like having nightmare of hands during my nights, indexing manually all those video frames. So fortunately now with computer vision and machine learning, fine-grained behavioral analysis can be more streamlined and automatized. Another big challenge is developmental. And so for instance, there is like the work of Vicky Leong or Stephanie Hall on recording babies in hyperscanning, and it's highly challenging because babies are not that easy to record. So that's a hope that we will get there and find more methods to get this developmental hyperscanning done. And to me, like one of the most interesting hopes for the future is to go to causal effect and hyperstimulation. So for instance, Giacomo Novembre has worked on this dual transcranial stimulation to actually force physiologically the synchronization and see how it affects behavior. And it's very interesting because now it's not anymore correlational. You can really probe the causality. And interestingly, in their paper, they illustrate well also one thing that was asked before for hyperscanning. So to get a control condition when you don't have a control condition is to do pseudo-randomization of dyads. So in their case, that's the kind of control that they are doing. So if you have an observation of interbrain synchronization and you want to try its robustness, it should collapse when you are scrambling the partners of the dyad. So that's one way of doing statistics of those hyper connectivity patterns. And last but not least, the beyond the dyad, which is also a big hope that Suzanne Dicker pioneered with her work on kids in teaching condition with class, and to have this recording of multiple brain would also allow to better understand this social embodiment that I was talking about earlier, and how we can see groups as one system instead of the sum of each individual separately. And ultimately, a big challenge, and that's the thing I will try to develop with my lab in Montreal of the social physiology for precision psychiatry, is to go in clinical populations. So there are very interesting work already in fMRI, for instance, Ida Bilek on bipolar disorder. And with Suzanne Dicker, we are already collaborating on Hyperscanning EGI for autism. So those are like the big hopes for the field. And because of this growing interest in hyperscanning, well, we wrote how to create your own hyperscanning system for a lot of different brand of EEG. So if you are interested in the technical aspect, there is this paper trying to delineate methodologically how to set up your own hyperscanning system. And today, thanks to the effort of students and with Suzanne Dicker as well, we managed to release our alpha version of the Python toolbox for hyperscanning analysis. So it was brought up at the last webinar that we don't have yet a pipeline for analysis. Well, now we cannot say it anymore because there is our HypePipe, which is like a Python pipeline for analysis and visualization of, for the moment, EEG and MEG hyperscanning. But to me, it's like a seed. And since it's a community-driven project, I hope people even attending to the webinar would be motivated to join forces to make a standardized and rigorous pipeline for doing hyperscanning analysis in the future. So thank you very much for your attention. Well, let's see what are the questions right now.
All right, thanks, Guillaume. It was really nice and interesting seeing again all of these things. And while examining your work, I also had a big pleasure to. I have a feeling that you are asking the right questions. You froze a bit. Do you hear me? Yes.
Yes. I think my computer is too old.
Yes, but you're used to working with old equipment, like the dual screen from 2010. I heard you took it from your basement.
Yes. So actually, that's sometimes old school analogic tools are really nice because when you use a digital camera, there is incompressible delay, while if you do optics and analogic, well, you are at the speed of light, so it's nice. There are very good papers historically. For instance, one of my favorite papers from Francisco Varela, it's not hyperscanning, but they are recording alpha activity in analog, and they are showing LED lighting up either in phase or anti-phase with the alpha waves. And because of the analogic, it's no delay. And by tweaking the phase or anti-phase, they managed to change the subjective experience of the light being light up or not for the person that is recorded. I found it super beautiful and a good example of how you don't need necessarily a gigantic setup, but more like a focused question and the right tool.
Yeah, you know that we are in the world of mobile EEG. So what I'm asking everyone is, how do you think that mobile EEG and advancements there will change the picture on hyperscanning studies?
Yeah, well, the work of Suzanne on that in school show how we can brought the lab, the neuroscience lab, in the context of people and not bringing the people in the neuroscience lab. And those mobile EEG are part of the answer methodologically. Then the key aspect is how we combine this very controlled experiment with a more ecological experiment. So for instance, in the 2010, we had these Faraday cages because we wanted to be sure that the interbrain measurements that we had was not caused by a joint source of electromagnetic interference, for instance, which is like something that is, well, if you do EEG in ecological context, there is a big challenge for artifact rejection and correction for those potential spurious source of synchronization.
That actually brings me to my next question. While I see that questions are piling up, we've been discussing lab streaming layer as a promising method for synchronizing not only EEG systems but also other systems like cameras, and it seems that this indeed would make possible to more labs to apply similar studies. What is your perspective on LSL?
So LSL, I think it's gonna be a very important tools to manage interoperability of different devices. Indeed, like we show here more like brain data, but and I briefly mentioned the video coding. So synchronizing video with motion capture or with physiological recording, that's more and more necessary. I didn't spoke about, there's another type of work I'm doing with virtual avatar, tracking ongoing movement and trying to regress representation of in the neural dynamics of ongoing behavior, really like a motion capture, so to say. These require milliseconds precision of the behavior. And for now, I honestly didn't try LSL yet, but that's the next thing I wanted to use for experiments. Yeah,
it's a very good tool. I personally used it when forming and participating neuroergonomics experiment and lab in 2014, and basically this allowed us to synchronize the Kinect and with EEG and everything. So it was very powerful, and I wouldn't know any other way to do it. I have a question from anonymous attendee. Is there a clear biological explanation for between brain synchronization?
So there are not a single one, but there are potential candidates, I would say. So let me, my computer is like super slow. Can I share? Whoa. So in 2012, I did a paper trying to simulate. Okay, I'm gonna share again my screen to get like up. Can you see that?
Yeah, I can see that.
So this is one big question, like what we are measuring when we are measuring this interbrain synchronization. And the thing is that there are many factors contributing to that. So first, the fact of being the same species and having a similar brain is making our dynamics look alike. So if you record two random number generator, they won't be synchronized, but that's extreme. if you record two people from the same environment and with the same neurobiology structure, it would tend to be more similar at the dynamical level. So is it like spurious or is it like relevant? Well, that depends on the scientific question. And indeed, proper design of the experiment allow you to contrast what is the baseline synchronization between the two brains. And in a way, that's also the hype part of hyperscanning I get as a reviewer often is like people are always surprised to find interbrain synchronization. Actually, the most surprising would to not find any, because of course, one human brain looks like another human brain. The importance is to really make the proper contrast from an experimental design point of view. And so beyond the neurobiological similarity, you have also the behavioral similarity. So to what extent you have more synchronization between two brains when they are doing the same task? And to what extent you have more interbrain synchronization if they are doing the same task in the same culture. So for instance, there is Kitayama and Andreas Ruppendorf work on cultural neuroscience and how, for instance, in Asia or in America or in Europe, you will not have necessarily the same patterns of social coordination. So culture is also part of these factors that you should account. And so that's more like the similarity aspect. And so the other phase that is the most interesting to me is the communication aspect. What kind of information is transmitted from one physiological system to another physiological system through the mean of perception and action. And in that case, you can have unidirectional flow of information or bidirectional flow of information. And those two things, the two different factors are connected because it's through communication and the reciprocal exchange of people that we are building our cultural similarity and our culture. And meanwhile, working on autism and all the neurodevelopmental condition, neurobiology and the similarity or dissimilarity of neurobiological grounding could impact your approach of communication with others. And so that's to show you one possibility of mechanisms of this interbrain synchronization. So in 2012, I did like a dyadic neurocomputational models of two brains in interaction using connectomics. And one of the conclusions, so the idea is to simulate two brains in interaction. And I passed on the technical part, but whoop. To make long story short, what's going on with those simulations is that the way you are shaped anatomically constrain your propensity to resonate with another person. And so like here in this graph, you have on the x-axis, the coupling between people, inter-individual coupling, C-inter, which is in the model quantify the flow of information from one virtual brain to another virtual brain. And on the y-axis, you have the hyperphase locking values of the interbrain connectivity. And what you see is that indeed, even in absence of communication of information, so when C-inter is 0, there is a spurious synchronization, even in the scrambled anatomy, because the system is not totally random. There's still a similarity intrinsically of dealing with similar physiological system. And when you compare the red and the green lines with the blue line, now you get the anatomy. So how the real structure of the human brain is already facilitating similarity compared to just random connections. And more interestingly, when you increase the coupling and the exchange of information, it doesn't increase in the red and the green line. So the random network doesn't potentiate interbrain synchronization, but the real connectivity in the blue line, in that case, you increase the interbrain synchronization if you increase the flow of information. So it means that the real interbrain synchronization that are interesting, so to say, are the ones that are between this point on the upper right and this point on the middle left. So the difference that you gain compared to baseline when you exchange information between those two systems. So that's one mechanisms of those interbrain synchronizations, so shared anatomy.
Actually, maybe that is an answer, and maybe not to the next question. What would you say has been the biggest discovery enabled by hyperscanning approach that would not have been possible without it?
Well, to me, like there is like political and scientific discoveries, like political in the sense that it brought up back in social neuroscience the fact that we were like totally looking where the light was for searching our keys, and we were like not at all looking at interactive paradigm. And so in that sense, the pioneering work of people in fMRI was also motivated by more neuroeconomics question, to me brought back this social interactive aspect in the game. And people like Leonard Schilbach, for instance, brought up the second person neuroscience, and it's part of this interactive turn of social neuroscience that is very important, and hyperscanning brought into it. To me, like one of the big results is the one with Pavel Goldstein on the affective touch, because it showed that you can achieve those interbrain synchronization without rhythmicity, because it's a touch. And it kind of cancel out the traditional criticism to say, oh yeah, but that's obvious that you get synchronization because you are doing the same rhythmic task on both sides. And with affective touch, we get also the emotional and affective dimension that is that we're missing. Yeah.
Yeah, that was one of my favorite discoveries you had. But I
helped for the analysis, but it's people from IFA who are really at the beginning for that, and hopefully also because we had discussion with Pavel on those aspects. That was also one of the very nice collaboration around hyperscanning that I had. Yeah.
Yeah, so there is a number of questions on your Hype now, and it comes at the right time because during the previous discussions we had, it was always the question, is there a pipeline? What is the framework? So yeah. I have one of my questions to add. As I mentioned, live streaming layer is a very promising approach to synchronize both multiple EEGs and other heterogeneous data. So do you plan to support .xdf file type in the Hype?
Well, it's community driven. So if people are interested by a format or a specific function, so it's all open source, and we want the community to build up around that. So we planted a seed with a lot of work last night actually to get it ready for the webinar with Suzanne and Anaëlle, Florence and Phoebe. And so the thing is, it's an alpha version, and we have also a preprint delineating a bit the roadmap of what kind of functions we see for iPipe. But those functions, we need help, and we need also ideas, feedback. So it's up to the community.
All right. So just not to spend the entire time that you have, there seems to be a desire for a Hype webinar, just for you to know. And
that could be nice, yeah.
A couple of questions on artifact-free epochs. Does the Hype have the ability to match the artifact-free epochs between dyads?
Yeah, so that's one of the part of the functions that we designed for this alpha version is to have ICA correction and automated artifact rejection and combination at the dyadic level. So this already started in the iPipe version right now. The idea is to, for now, keep only the non-artifacted period for both subjects or more subjects. Because that's one of the pain of hyperscanning is indeed that when it's not one subject that blinks, it's the other, so you lose a lot of data.
Actually, I have a question on that part. Did you try artifact subspace reconstruction ASR from SCCN for the artifact rejection?
No, we didn't try that yet. We have just like automated detection of bad channels thanks to a wonderful library called auto-reject. So, and actually that's important to mention that iPipe is built as a kind of extension for MNE Python, which is like the biggest Python library for MEG and EEG. So we are deeply thankful and grateful for MNE Python to exist because it saves a lot of time, because when you get your data into MNE, then iPipe handle the structure from the MNE side. And so like this automated rejection techniques is from another extension of MNE called auto-reject. But I can see that as really a community sandbox. So if someone has a new idea of algorithm or rejection and so on, that's all for it.
Well, from our perspective, we had a chance to showcase ASR last year on the mobile EEG workshop in Belgrade. I would add that it's a very, very powerful tool, and I think that people who didn't see it are kind of fascinated always, at least in my experience. And we might get it out there also as alpha version in the software, so people can use it live, let's say.
Yeah, and actually I see comment from Suzanne that indeed one of the things is that we allow also people to load their own preprocessed data in iPipe. So it's not, you can handle your preprocessing somewhere else and then get it into iPipe. Yeah,
that's good. That's good to know. Okay. So
I see many other
there are many other questions.
So I can try to answer quickly. So the first one is about frequency band in the same brain area between subject and not in other frequency. What is your theoretical explanation from a dynamical system approach? Well, that's a very good question. So indeed, there is known now through RTMS that certain brain region resonates in different frequency. So there is a gradient of resonance, and especially right parietal resonate in alpha frequency. So it's not surprising to me to find the strongest interbrain synchronization in the alpha in the right parietal. So there are like dynamical reason for that. Then there is also a functional aspect to that, because in our last paper with more virtual avatar, we show that RTPJ is also a kind of a hub between self and other treatment of information. So I think dynamical system approach can lead to mechanisms, but ultimately we need also a functional interpretation of what's happened with this interbrain synchronization. Then I see another question. What is the biggest constraint when conducting a study with hyperscanning? I think it's like the recruitment and the logistic and the synchronization. Once you have the data, you're fine, but the data collection is it's really a big constraint logistically. And then, of course, as I said, like to have a clear question with the proper design. But that's more like to me, it's the the enjoyable part, not the pain part is really the logistics and so on. Then I see a question about multiscale analysis. And I see networks of networks. So that's actually discussed with Suzanne, especially for the group hyperscanning. So how to have a nested network of networks.
Maybe read the full question before you answer, so we can maybe post it later for people to search.
Sure, sorry. Then there is like, I was curious about your next steps in terms of research question using hyperscanning after moving to Montreal. So as I say, like one of the motivation is really to introduce this social physiology into psychiatry and how we can go from the molecular level to the social level. And actually we are even working with brilliant young psychiatrists like Christophe Go, who's maybe there, on how social consensus even shape our objects for studying social psychopathology. So the symptoms networks, for instance. And so it's really to connect this social dimension with the biological grounding. And so hyperscanning is part of the tools, but it's not like, I don't want to create a lab about hyperscanning. It's more like it's part of the palette of tools that are gonna be used. Yeah.
If I can make it just a small break, I have my usual poll time. I want to ask people who are around, what do they think, and do they consider using hyperscanning in their future EEG projects? So let's. I would ask people who are here with us to vote and see what's going on.
So I'm reading the next questions.
Sure.
So there's a question by Juan about any thought on how to deal with potential confound of having the pair of subject coupled to the same environment, generating hyperconnectivity relate to this coupling instead of result for the interaction itself. So I kind of answered already that with my double similarity communication, but this partially addressed by the design you are doing and how you contrast condition where the environment, the shared environment is common to the two conditions, so that by contrasting you remove this shared effect of the environment. Then
seems that the poll, I'm going to share it. Yeah. So let's say a lot of people who are here looking at you are already using, and another half is interested in that. So here is a nice thing to know. I just want to add that if any of you think that mobile EEG is the way to go with your future hyperscanning studies, you can actually get in touch with MBT. You think we can assist on that? I'm going to share in the a link in the chat window where you can schedule a talk with us. You should get it. All right, Guillaume, I'm back to you. What's the next question?
Yeah, so next question. So there is, would the IP work to analyze hyperscanning EEG data from parent-child or parent-infant dyad? Yes, that's actually already the case. And the most complicated aspect is to put your data into MNE, but once you have your data into the MNE Python structure, it should be fine. So it's not like really the source of the data that is problematic, but more like at least the format, depending on certain format of EEG. But MNE is really an amazing toolbox in Python to get most of the format in there. A little trick, actually, I see also the one question about, would the toolbox be translated in MATLAB by Quentin Moreau? I don't think so, because I moved away from MATLAB to get full open source in Python. But one positive aspect about MATLAB is that the EEG lab toolbox actually is older than MNE and under a lot of different format for EEG. And so one trick is if you have MATLAB to load your data into EEG Lab and then save in the EEG Lab format. So yeah, you can use that to get it into MNE and iPipe. So we are still dependent for some arcane and rare EEG format to go through EEG Lab and MATLAB. Yeah. Then what else? I'm not of the field. What are possible stimuli that would be used to create such resonation with another person that then we can analyze? So that's not a stupid question, actually. It's a very important question, Sophia. And actually, you can use many. I show, for instance, in the case of Ureson, they use even cinema as a joint stimulation. There's not so much exchange between people, but someone like Giacomo Novembre working a lot on music. Music is a wonderful sandbox for studying social interaction and interaction with stimulation at the perceptual level, because something shared and that can be also played together. And also what we saw with the work of Susan Dicker is learning. Like a teacher is a wonderful stimulus in a way, a very complex and useful one. So yeah.
And
then,
actually, this. Sorry for interrupting you,
yeah,
but this is something that you mentioned in one of your previous talks and related to your paper from 2014 about how we use how we do learning today. It's sort of a one way, and your hyperscanning results in a way suggest that might be a more optimal component or way to adapt the teaching and learning. Would you care to comment on that?
Yeah, so that connect a bit indeed with especially what I mentioned with the replication in MRI of my work with the team of Philippe Fossati with Sophie Guillaume, when they found that you have a intrinsic reward in spontaneous interaction that is not present in the forced interaction. And on that, I wrote 2 papers with Jan Clark with more educational science about the consequences for peer learning and how actually teaching in a more collaborative way would be more rewarding. Well, I mean, sometimes neuroscience brought like something that seems obvious for educator and so on. But one of the takeaway message is that like unidirectional teaching style is not rewarding intrinsically, while this more like peer learning, collaborative teaching style could be and could help in having a more efficient teaching experience. Yeah.
All right. So
I have 3 last.
More.
Yeah, 3 last questions. They keep coming.
They keep coming. So you have to answer fast because you know. This
so
is
there
feedback.
Is one by Mitra. Is hyperscanning suitable for time perception study? Why not? Again, ask yourself if you really need hyperscanning to answer your question. But it could be, for instance, interesting to see if 2 people embedded in the same social interaction would have the same estimation of the time passing, especially with the turn taking and so on. That could be one things to explore, maybe. Another question from, yes?
I can answer this one from Juan. So can you do hyperscanning with a single EEG amplifier, e.g. using a split ground cable?
Yes.
I don't find it practical due to the, especially the ground and referencing issues. It's even the case with some high differences. For instance, if you're trying to use the same amplifier for EEG and EMG, and the reference can go wild. So it's not really advisable, although it may be possible, but not. You should definitely not rely on single amplifiers for that.
That can be tricky, yeah, for the reference.
Yeah.
For another question from Quentin Moreau. Is cross-frequency coupling between brains a potential future direction for hyperscanning analysis? Yes, it's part of actually the roadmap for HyPipe to help digging into this kind of more complex connectivity measure. Again, it's what would be the question. So for one question coming from the result I show with the in 2010 with the parietal being synchronized in alpha and going more frontal in the gamma. There is now growing level of interest in theta gamma cross-frequency coupling. In EG, it's gonna be hard because gamma in EG is tricky, but in dual MEG, that could be the kind of thing you want to look at. So to what extent, for instance, the theta of the ongoing movement of one subject would nest the gamma of the other subjects in the prefrontal, for instance. That could be done actually. In MEG, I think it would be easier to approach. And there is like 2 MEG in Montreal, so I hope we're gonna make it.
Okay.
And the last question, what body tracking tool would you recommend for behavioral measure today? Wow. So I started actually my first internship in science using motion capture at the Collège de France in Paris. At that time, it was with the Vicon system, which is super expensive and very nice. It's like for cinema. Now I'm more like open source do-it-yourself approach. So actually with Arduino and a couple of good sensor, you can have cheap and still reliable measurements. At the intermediate level, I'm playing also with something called Leap Motion, which is like $100 kind of hand tracking. And it's not that expensive and it allows to track simple movement of the hands. I mean, the trendy thing also right now is to use just a webcam with deep lab cut. So there's like this deep learning computer vision toolbox that with the webcam can reconstruct your bones and the structure of someone dancing, etc. It's widely used now in neuroscience, and I think that's also something that can be hacked into experimental design. Yeah.
Yeah, as I said, we also used a couple of years ago, and there are a few papers. I don't know if I mentioned them to you. We also used Kinect for body tracking. We used Leap Motion, but our data was not really, like the sensor kept losing the hands, and we ended up not including it in the overall analysis. But indeed, it's a very nice and cheap tool to achieve those things. From our end, we are trying to kind of gather around the people who are in hyperscanning to create a community that would share the information like the ones that you just shared with us about the Python pipeline, which is I think very good. I'm going to share my screen again just to end this. This thing. So we're going to place this today's talk on our YouTube channel, completely or partially. Probably we'll cut some questions so that that they could be searched directly online. And we have a Discord channel on hyperscanning. I think this is going to be very nice for the community. We'll share that with participants. And as I said, if people are considering mobile EEG for hyperscanning, they can schedule. You can schedule a meeting with us. I just shared a link in the chat window, and I would end with one of my last questions there, which is maybe stupid. So it's now my time to ask stupid questions, but it's intriguing, like the the model and imitator study that you kind of started from. Why do you think these studies are relevant? Why is it something very inherent in our everyday behavior, or and how does it affect what we do?
Yeah, so the idea of looking at imitation came from the developmental psychology perspective, because before getting to language, we are communicating through movements. And one of the core scientific questions I'm interested in is how representation of others and complex symbols like language, etc., are actually not dissociated from asymbolic, nonverbal, movement-based communication. And it was also pragmatic because language is a tricky beast for artifacts and so on. But I coming from more embodied cognition and Varela's lab and this sensory motor and active account, and now progressively getting back to language, representation, attribution of intention, and so on. But it's a path, like wanderer, there is no path, the path is made by walking.
This was very interesting for me, Guillaume, and thank you very much for sharing.
Thank you for the invitation.
And I hope that we see each other soon, and that I at least have the chance to visit your new lab. And wish you best of luck. So in your future work, and looking forward to seeing it. And thanks everyone for being with us, and see you next time.
Bye-bye, and take care.
Take care.